The segment for genomic medicine today is on the 9p21 story. This is really a fascinating one, which goes back to June of 2007. At that time, both the Icelandic group, with multiple cohorts from the U.S. and elsewhere, along with an Ottawa, Canada group with many collaborating groups in Dallas and throughout Europe, found the 9p21 signal, which is a single nucleotide polymorphism at this locus being highly correlated with coronary disease and myocardial infarction. Since that time, many other groups have confirmed the 9p21 association with coronary disease, but this particular interesting SNP is not in a gene, and in fact is hundreds of thousands of bases away from the closest gene, which is CDKN2B, a gene that's known for tumor suppression, cell cycle proliferation, senescence, and aging. Not particularly clearly associated with heart attack. And interestingly, another SNP in the same portion of the genome in 9p21 was in June last year identified to be associated with diabetes. And in fact, there was a lot of speculation at that time that these two SNPs, being less than 10,000 base pairs away, were indeed extraordinarily important, and the two diseases had converged. But in fact, that wasn't the case. And the reason that wasn't the case, and the extension of this important work, comes from a new paper in Nature Genetics, published online in January 2008. This again comes chiefly from the DECODE group, from Carrie Stephenson and colleagues. And basically what it did was a very interesting study involving many different cohorts throughout Europe, including not only coronary disease and myocardial infarction, diabetes, but also the new vascular phenotypes, particularly of abdominal aortic aneurysm and intracranial aneurysm, that is the Berry aneurysm. And the main conclusions from this study were twofold. First, that there were the two SNPs in 9p21 are clearly separate and distinct, and there is no evidence for convergence. That is, that the diabetes SNP was in no way associated with the vascular phenotypes, but indeed that confirmation of diabetes associated with 9p21 was indeed confirmed. On the other hand, the new big extension of this work from looking at other vascular phenotypes was that abdominal aortic aneurysm, as well as intracranial aneurysm, which is non-atherosclerotic, were highly associated with the same SNP in 9p21. And in fact, this particular SNP, the odds ratio is for heterozygotes is 1.3 for each of those four phenotypes, whether it's AAA, intracranial aneurysm, MI, and coronary artery disease. Now, what was interesting is that peripheral artery disease and a proxy for cerebrovascular disease came in at a lower level. 1.15 was the odds ratio for heterozygotes. And it wasn't as impressive, but certainly going in the same direction, not quite the same magnitude. And it would be premature to think that a particular variant would predict for coronary and abdominal aortic aneurysm and a non-atherosclerotic disease, Berry aneurysm, but not have some association with peripheral artery disease or cerebrovascular atherosclerotic disease. So my sense about this is that it may well be a vascular phenotype, but it's not just atherosclerosis. And it invokes the likelihood of a vessel wall defect, particularly immediate defect, which is what is absent in Berry aneurysms. So it now appears that we have, for the first time ever, a marker which turns out to be genomic, which is a marker for two diseases that we never had a marker diagnostic before, which is AAA and intracranial aneurysm, both of which can be fatal. Berry aneurysm rupture associated with subarachnoid bleed is quite important, if we could find these individuals before they ever have such an event. And an abdominal aortic aneurysm is a very difficult thing to diagnose, except when there's a abdominal aortic aneurysm pulsatile on exam, which is unusual with bruits or associated with peripheral vascular disease, but oftentimes it is missed. So we have a new marker and a whole new thinking about what 9p21 means. There's two SNPs in there. One's clearly related to diabetes. It's very separate and does not co-segregate with the SNP, which is a vascular phenotype 1, even extending outside of atherosclerosis. Should these new phenotypes of AAA and intracranial aneurysm be looked at for those who have homozygous variants in 9p21? This is an intriguing question. Should patients who are homozygous for the variant undergo ultrasound aortic distal segment to see whether there's aneurysm, or magnetic resonance angiography to see whether or not they have a barrianeurysm? These are new questions that are coming up as a result of this very interesting study showing the pleiotropic aspects of 9p21. So I'd be really interested in your thoughts, and please post your ideas about how to process this fascinating new data on the Genomic Medicine website. Thanks for your attention.